Bismillahirrahmanirrahim. Our next topic from unit number 4, cell structure and function, is to give the outline of structures and functions of some given cellular organelles. One of them is nucleus, other one is endoplasmic reticulum, Golgi apparatus, and mitochondria. So let's start with the proceeding. So these are the few given organelles from the eukaryotic cells. So the question is, why do eukaryotic cells contain endomembranous organelles? You know that eukaryotic cells are often larger and more complicated than prokaryotic cells, and as cells got larger during evolution, simple diffusion, random movement of molecules within them was not able to carry out all the life activities. So, in order to overcome this problem, eukaryotic cells have evolved endomembranous organelles, mean membrane-bounded compartments within the cell that are individually specialized for certain specialized functions. What are the key reasons to have this sort of organelles in and possess these sort of organelles in eukaryotic cells? Number one is the compartmentalization. Different biochemical reactions can happen simultaneously without interfering with each other. For example, digestion in lysosome won't damage all other cellular cell structures, because in case of in case of an animal cell, what will happen that the cells whose function is to perform the digestion, that lysosome will be having more dangerous, more hydrolytic enzymes, so they should be confined with that particular lysosome structure. Otherwise, rest of the cellular areas or structure may get damaged. So this compartmentalization is important to keep things separate from one another. Then comes the efficiency and organization. Specialized environment allows enzymes to work at their ideal condition. For example, they need special pH and ionic conditions, faster and more controlled transport of molecules, because we know that in case of endoplasmic reticulum, there is vesicle formation, which that sort of vesicles, they will get entry into the Golgi complex area, and they get merged with the uh, the Christie, rather cisternae of the the Golgi complex. Then the increased surface area. So membranes provide more surface area for essential processes. For example, ATP synthesis on inner mitochondrial membrane. And then the complex function. Eukaryotic cells carry out complex processes like protein modification. For example, the functioning of Golgi apparatus, lipid synthesis in case of smooth endoplasmic reticulum, and energy production in case of mitochondria. They need dedicated separate spaces. And then the intracellular transport, the endomembranous system, for example, endoplasmic reticulum, Golgi vesicles, they help to move proteins, lipids, and other substances precisely where they need it, inside or outside of the cell. So eukaryotic cells have organelles to work efficiently and stay organized. Now, we will start with the given organelles in our syllabus. The first one and the most important one is nucleus. We know that the nucleus main function is to control the cell function, cell activities. So it is a control cell center of the cell. And secondly, usually structure-wise, it is the largest organelle and function to there are some function assigned to the nucleus. For example, to store the genetic information in the form of DNA, control cell growth, metabolism, reproduction, means that each and every activity of the cell is being controlled by the activity of nucleus. What are the parts of nucleus? Nuclear envelope, which is double-layered membrane surrounding the nucleus, has nuclear pores that allow substances like RNA and ribosomes to move in and out. Obviously, RNA, messenger RNA, ribosome, they will go out of the nucleus, and other bigger molecules may get entry inside the nucleus to perform their function. Nucleoplasm, semi-fluid substance inside the nucleus, just like the cytoplasm, contains chromatin and nucleolus, contains many enzymes and dissolved molecules. Chromatin, a mix of DNA and proteins, histones. DNA stores genetic instructions. During cell division, chromatin condenses into visible chromosomes. So nucleolus, dark, dense area inside the nucleus, and its function is to, it is responsible for the production of ribosomal RNA, assembles ribosomes, which later move to the cytoplasm to help in protein synthesis. Chromosomes. So the condensed form of chromatin material is will form chromosomes. So structure made of DNA and proteins carry genes which determine traits and control cell activities. Chromosome appear as thread-like structure during cell division, whether it is mitosis or meiosis. So this is sort of diagrammatic depiction of the nucleus. So here the nuclear envelope, nuclear pores, DNA in the form of chromatin material, and nucleolus. So that is all about nucleus. Then comes the endoplasmic reticulum, a network of membranes form channels throughout the cytoplasm, often connected to the nuclear envelope. There are two types: rough endoplasmic reticulum, RER, covered with ribosomes, looks rough under the microscope. That is why it is rough endoplasmic reticulum. Function is to synthesize proteins, modifies proteins, add sugar chains, transport proteins to the Golgi apparatus for further processes. Smooth endoplasmic reticulum lacks ribosomes, so that's why it looks smooth under microscope. The functions assigned to it, first one is to make lipid, fats, including steroid hormones, detoxify drugs and harmful substances. For example, in case of liver area, the liver having liver cells having smooth endoplasmic reticulum, they are supposed to detoxify the harmful substances and drugs. Store calcium in muscle cells, helping the muscle contraction, helps transport material within the cell, provide mechanical support to help maintain the cell shape. So this is a sort of a sketch of smooth and the rough endoplasmic reticulum. The endoplasmic reticulum is in continuity with the nuclear membrane, then rough endoplasmic reticulum due to ribosomes, and the smooth endoplasmic reticulum is without ribosomes. The other organelle is Golgi apparatus, also named as Golgi body, Golgi complex in case of plants dictyosome. So discovered by Camillo Golgi in 1989, 98, and made up of flattened stacked membrane sacs called cisternae. So keep in mind that individual tube-like structure in case of endoplasmic reticulum, and in case of the Golgi apparatus, they are named as cisternae. Cisterna and plural is cisternae. So their function is, the function of Golgi apparatus is to receive proteins and lipids from ER, endoplasmic reticulum, mostly smooth, modifies these molecules, may add carbohydrates to make glycoproteins or glycolipids, package material into vesicles for transport within the cell or out of the cell if that cell is meant for the production of certain secretions like enzymes in case of pancreas and like that, and through exocytosis. In cells that secrete substances, the Golgi apparatus, they are well developed. So Golgi structure in detail has two phases: forming phase, also named as cis phase, where vesicles from ER arrives and fuse with the cisternae; maturing phase, also named as trans phase, where new vesicles budded off and move towards toward their destiny, whether within the same cell or out for the export. So these are the cisternae. As I told you that individual tube is cisterna and plural is cisternae. And inside the lumen activities are being carried out by certain enzymes. So incoming transport vesicles, cis forming phase, and outgoing transforming phase. These vesicles are secretory vesicles, and they may go out of the cell if required. Then the last organelle given in our syllabus is mitochondria, known as the powerhouse of the cell. We know since our childhood because they produce energy, found in large numbers in cells, needing lots of energy. For example, muscle cell, nerve cells, sperm cells, ovum cell, and their shape often rod shape, oval or thread like, depending upon the cell in which they are. Structure, it's a double membrane structure. Outer membrane smooth, contains channels, porins, allowing small molecules to pass through. Inner membrane highly folded into structure called cristae. So this is the difference. Crista or plural cristae is the finger-like projection in case of mitochondria, and they should be different from cisternae of the Golgi complex and the endoplasmic reticulum. We should remember it because it's very tricky to separate them. So cristae increase the surface area for energy production. Mitochondrial matrix, fluid-filled space inside the inner membrane, contains enzyme for respiration, Krebs cycle enzymes, ribosomes for protein synthesis, circular mitochondrial DNA, empty DNA. So it is what we say semi-autonomous organelle. So mitochondria can produce its own protein. So it is supporting the presence of chloroplast and mitochondria are the supporting evidence in favor of endomembranous theory of eukaryotic cells formation. So what is the function of mitochondria? Perform aerobic respiration, turning nutrients into energy. That nutrients mean the energy molecules, the macromolecules whose function is to produce energy. And they will produce energy in the form ATP, in the form of ATP. It can store calcium ion and help regulate their level in cells, help to produce heat in special tissues, for example, brown fat, without the production of ATP and releasing energy and keeping the animal and that particular organism the homothermic. So this is the detailed structure of mitochondria, outer membrane and the inner membrane having projections. These sort of projection, one is crista and plural cristae. Inside the matrix, granules are there and ribosomes are there. The nuclear DNA is there. So these sort of structures, they are meant for their special functions. So what are the advantages to have these sort of organelles? Number one, compartmentalization helps the cell to perform their function efficiently, allows specific reaction to happen in dedicated areas, specialized areas, keep harmful substances safely enclosed, maintains the different conditions, for example, pH required for certain activities, make it easier for materials to transport around the cell. So quick summary of the functions of the given organelles. Nucleus store DNA, controls activities, all activities, makes ribosomes, RER, makes and process proteins, smooth endoplasmic reticulum makes lipid, detoxifies certain drugs, store calcium. Golgi apparatus modifies and packages proteins or lipids for uh, inside transport or outside of the cell. Mitochondria produce ATP energy and regulate cell metabolism. So these are the different organelles given in our syllabus. That is all from my side. So Allah Hafiz.